You are here because you are tired. Not the tired that comes from physical labor. Not the tired that a single night of sleep can cure. But a deeper exhaustion. One that lives in the architecture of your nervous system. Your brain. That 3-pound universe of electrical activity has been running at a frequency it was never designed to maintain. And tonight, we will understand why. Let us begin with a simple biological truth. Your brain operates on chemicals, neurotransmitters, those molecular messengers crossing the microscopic gaps between neurons, creating every thought, every sensation, every moment of your conscious experience. Among these chemicals, dopamine holds a particular position. It is not, as popular culture suggests, simply the pleasure molecule. Dopamine is the neurotransmitter of motivation, of prediction, of the anticipation of reward. It is the chemical voice that whispers, something important might happen next. In the ancestral environment, dopamine served survival, the rustle in the grass, the unexpected movement at the edge of vision, the possibility of food or danger. These moments triggered dopamine release, sharpening focus, heightening awareness, preparing the organism for action. The system was designed for intermittent activation, moments of heightened alertness, followed by long periods of baseline calm. But the modern environment, the world of screens and notifications, has created something unprecedented in human history. A state of continuous dopamine activation. Consider what happened today. Before you even left your bed, you reached for the device. The screen illuminated, and within seconds, your brain began processing dozens of distinct stimuli. A notification indicating social validation, a headline suggesting threat or novelty, an image calibrated through algorithmic precision to capture attention, a video edited at a pace faster than natural human conversation. Each of these inputs, each distinct piece of information, triggered a small release of dopamine. Not the large surge of genuine reward, but micro doses, rapid fire, creating a pattern of stimulation your nervous system interprets as perpetual anticipation. Something important might happen next, and next, and next. The problem is not the dopamine itself. The problem is the frequency. Your neurons, those delicate cellular structures, operate on principles of electrochemical balance. When dopamine is released, it crosses the synaptic cleft, that infinitesimal space between neurons, and binds to receptors on the receiving cell. These receptors are protein structures embedded in the cell membrane, shaped with molecular precision to recognize dopamine and dopamine alone. When binding occurs, the receptor changes configuration, triggering a cascade of intracellular events, ultimately generating an electrical signal. This is how information travels through your brain. Chemistry becomes electricity. Electricity becomes thought. But here is what the biological textbooks often fail to emphasize. This system was engineered for dynamic range, for contrast, for the ability to detect signal against background noise. When dopamine release becomes continuous, when the receptors are constantly occupied, the nervous system faces a fundamental problem. It cannot distinguish meaningful signal from constant static. The brain, in its ancient wisdom, begins to adapt. This adaptation has a name: downregulation. The neurons receiving constant dopamine input begin to reduce the number of available receptors on their surface. Think of it as a biological defense mechanism, like pupils constricting in bright light, or ears adjusting to prolonged noise. The neurons are attempting to restore balance. But this creates a paradox. The more stimulation you seek, the less capable you become of feeling stimulated. You need more input to feel less response. The threshold for pleasure, for motivation, for the sense that something matters, rises steadily higher, and so you scroll faster. You switch between applications more rapidly. You seek content with higher velocity, brighter colors, more aggressive editing, louder audio. You are not weak. You are not lacking discipline. You are experiencing a predictable neurochemical response to environmental conditions that did not exist 1 generation ago. Your brain is trying to protect itself by becoming numb. Let us examine the electrical signature of this state. Your brain generates measurable electrical activity, patterns of synchronized firing across millions of neurons. These patterns, these brain waves, can be classified by frequency. Delta waves, the slowest, occurring during deep sleep, measured at 0.5 to 4 cycles per second. Theta waves, the realm of meditation and light sleep, 4 to 8 cycles per second. Alpha waves, calm wakefulness, relaxed attention, 8 to 12 cycles per second. Beta waves, active thinking and focus, 12 to 30 cycles per second. And then, there is high beta, the frequency of anxiety, of hyper-vigilance, of the mind that cannot stop processing, 25 to 35 cycles per second, sometimes higher. When you are scrolling through the endless feed, when you are switching between email and messages and news and video, your prefrontal cortex is locked in high beta. This is not the productive focus of deep work. This is the scattered attention of threat assessment. Your brain is trying to track multiple streams of potentially important information simultaneously. It is exhausting because the prefrontal cortex, that most recently evolved region of your brain, responsible for executive function and impulse control and long-term planning, requires tremendous metabolic energy. It consumes glucose and oxygen at rates far exceeding its size, and when it operates in high beta for hours, for days, for weeks, it depletes its reserves. The fatigue you feel is real. It is not psychological weakness. It is metabolic depletion. Your neurons have been firing at maximum capacity, burning through adenosine triphosphate, that cellular currency of energy, faster than mitochondria can replenish it. The synapses, those connection points between neurons, accumulate metabolic waste products, proteins that need to be cleared, damaged cellular components that need to be recycled. But clearance requires rest. It requires the brain to shift out of high beta, to descend through beta, to settle into alpha, and eventually to drop into the slow waves of theta and delta. This is what sleep is for. Not just the passive absence of wakefulness, but an active biological process of restoration. But sleep has become difficult because the dopamine system and the arousal system are intimately connected. When dopamine receptors are chronically stimulated, when the brain has adapted through downregulation, the entire nervous system remains in a state of heightened activation. Even when you close your eyes, even when you lie in darkness, the electrical activity continues. The mind generates thoughts, worries, plans, replays of conversations, anticipation of tomorrow's demands. You are tired, but you cannot rest. You are exhausted, but you cannot sleep. This is the signature of a nervous system caught in dysregulation. Let us be clear about what is happening at the cellular level. Your neurons communicate through both electrical and chemical signals. The electrical component, the action potential, travels down the axon at speeds up to 120 meters per second. But the chemical component, the release and reception of neurotransmitters, is slower, more nuanced, more susceptible to modulation. When you experience something genuinely rewarding, a conversation with someone you love, a moment of creative insight, a beautiful sunset, dopamine is released in the ventral tegmental area and the substantia nigra. These are clusters of neurons deep in the midbrain, ancient structures present in all mammals. The dopamine travels to the nucleus accumbens, to the prefrontal cortex, to the amygdala. It modulates activity across multiple brain regions, creating the integrated experience of reward and motivation. But the artificial stimulation from screens operates differently. It triggers dopamine release through novelty, through unpredictability, through the variable reward schedule that slot machines use. You do not know what the next scroll will reveal. Sometimes it is boring, sometimes it is surprising, sometimes it is outrageous. This unpredictability is neurochemically potent. Your brain releases dopamine not in response to the reward itself, but in response to the uncertainty. The possibility that something interesting might appear keeps you engaged, keeps you searching, keeps the dopamine system activated. This is why you can scroll for an hour and feel both stimulated and empty. You received constant microdoses of anticipation, but very little genuine reward. The consequences extend beyond dopamine. When the nervous system remains in high beta, when the prefrontal cortex is constantly active, other neurotransmitter systems become imbalanced. Norepinephrine, the chemical of alertness and stress, remains elevated. Cortisol, the hormone of sustained stress response, circulates at levels meant for short-term threats. Serotonin, which helps regulate mood and promote calm, becomes depleted in its storage sites. GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter, struggles to counteract the excitatory signals. Your nervous system becomes like an orchestra where every instrument is playing fortissimo, with no rests, no dynamic variation, no silence. The result is not music, it is noise. And you, the conscious awareness arising from this neural activity, experience this as mental fatigue, as difficulty concentrating, as the sense that nothing feels quite satisfying, as the inability to be present, because the mind is always anticipating the next input. This is not a moral failing. This is biology responding to environment. But here is what matters tonight. Here is why you are listening to these words. The same biological systems that adapted to overstimulation can adapt back. The neurons that reduced their dopamine receptors can increase them again. The prefrontal cortex that depleted its metabolic reserves can restore them. The brain waves that locked into high beta can descend into alpha, into theta, into the deep delta of restorative sleep. Your nervous system wants to heal. It has mechanisms, ancient and reliable, for returning to balance. But it needs something from you. It needs time. It needs absence of stimulation. It needs neural silence. This is what we mean by dopamine restoration. Not the elimination of dopamine, but the restoration of dynamic range, the return of contrast, the recovery of the ability to feel subtle rewards, to experience genuine motivation, to find pleasure in simple things. This happens through a process called upregulation, the mirror image of what occurred during overstimulation. When dopamine input decreases, when the constant barrage of novelty stops, the neurons begin to increase their receptor density again. They manufacture new receptor proteins. They transport them to the cell surface. They restore the sensitivity that was lost. But this process takes time. Days, not hours. The first 24 hours are the most difficult. Your brain, accustomed to constant stimulation, will generate powerful urges to seek it again. Reach for the phone, check the notification, open the app, begin the scroll. These are not conscious decisions. These are subcortical drives, impulses arising from brain regions that operate below awareness. The nucleus accumbens, desperate for dopamine, sends signals to the motor cortex. The hand moves before the conscious mind registers intention. This is why willpower alone often fails. You are not fighting against thought. You are fighting against neurochemistry. But you do not need to fight. You simply need to understand. When the urge arises, when the craving manifests, recognize it for what it is: a signal from neurons that have adapted to artificial stimulation and are now requesting it. The urge is not you. It is brain activity, chemistry, and electricity. And like all patterns of neural activity, it will pass. Typically within 10 to 20 minutes, if you do not reinforce it. What happens during those minutes is fascinating. The urge, the craving, manifests as increased activity in the anterior cingulate cortex. This region monitors for conflict between desires and goals. It generates the psychological tension you experience as temptation. But if you simply observe without acting, the activity begins to decrease. The prefrontal cortex, when not depleted, can modulate the subcortical drives. It can delay gratification. It can choose long-term benefit over short-term relief. This is not superhuman willpower. This is the ordinary function of a well-rested brain. And rest is what we are facilitating now. As you listen to these words, as your attention follows the slow rhythm of this explanation, your brain waves are beginning to shift. The high beta frequency, that rapid oscillation of hyper-vigilance, is starting to slow. Neural populations that were firing independently in chaotic patterns are beginning to synchronize. This synchronization, this coherent rhythm, is the signature of alpha waves, 8 to 12 cycles per second. The frequency of calm alertness, the state where you are awake, aware, but not striving. Alpha waves arise naturally when sensory input decreases, when you close your eyes, when you sit in quiet, when you listen to a single voice speaking at a measured pace. The thalamus, that relay station for sensory information, reduces its output to the cortex. The cortex, no longer bombarded with data, can settle into its intrinsic rhythms. Networks that were competing for attention begin to quiet. The default mode network, active during rest and internal reflection, becomes more prominent. You shift from external focus to internal awareness. This is the beginning of restoration, not the dramatic transformation, not the sudden cure, but the first step. The recognition that your nervous system can exist in a different state, that the high beta urgency is not permanent, that beneath the noise, there is a quieter frequency. Your brain remembers how to rest. It simply needs permission and practice. The dopamine system is beginning to adjust. In the ventral tegmental area, neurons that were firing constantly begin to return to tonic activity. Tonic, meaning baseline, the low-level firing that maintains readiness without exhaustion. The prefrontal cortex, relieved of constant decision-making and impulse control, begins to reduce its metabolic demand. Blood flow decreases slightly, oxygen consumption drops, glucose uptake slows. This is not shutdown. This is efficiency. The brain entering a lower power state while maintaining full capability. Like a computer dimming its screen to conserve energy while keeping all programs running. You may notice physical sensations as this occurs. A subtle heaviness in your limbs as muscles release chronic tension. A deepening of breath as the diaphragm no longer needs to support high arousal respiration. A softening around your eyes as the muscles that hold your gaze in sharp focus relax. These are not suggestions. These are physiological consequences of descending from high beta into alpha. The parasympathetic nervous system, the rest and digest branch, begins to gain influence. Heart rate variability increases, indicating flexibility in the autonomic nervous system. Saliva production increases. Digestion resumes normal function. Immune activity optimizes. Your body, like your brain, is designed to heal during rest. But the mind may resist. Thoughts may arise. Worries about tomorrow. Replays of today. Sudden memories of tasks undone. This is normal. The prefrontal cortex, accustomed to planning and problem solving, generates these thoughts automatically. They are not important. They are neural habit. Each thought, each worry, each plan is simply electrical activity in specific brain regions. The dorsolateral prefrontal cortex, the anterior cingulate, the hippocampus retrieving memories. You do not need to engage with them. You do not need to solve the problems they present. You simply notice them, acknowledge them, and return attention to the present moment, to the sound of this voice, to the rhythm of your breath, to the sensation of your body at rest. This practice, this gentle redirection of attention, is training the brain. You are strengthening the connections between the prefrontal cortex and the limbic system, specifically between the ventromedial prefrontal cortex and the amygdala. This pathway, when well developed, allows you to regulate emotional responses, to experience an urge and not immediately act, to notice anxiety and not spiral into worry, to feel restless and remain still. This is not suppression. This is regulation. The mature function of a nervous system that has options. And with each moment of rest, with each breath taken without urgency, your brain is rebuilding those options. The synapses that were depleted are replenishing their stores of neurotransmitters. The mitochondria in neurons are producing fresh ATP molecules. The glial cells, those support cells that outnumber neurons, are clearing metabolic debris. Microglia, the immune cells of the brain, are pruning damaged synapses and supporting healthy ones. Astrocytes, those star-shaped cells, are maintaining the blood-brain barrier and regulating neurotransmitter levels. Oligodendrocytes are maintaining the myelin sheaths that insulate axons and allow rapid signal transmission. All of this is happening right now. Your brain is not passive. It is a living system, constantly maintaining itself, constantly adapting, constantly seeking equilibrium. And equilibrium is what we call homeostasis, the biological tendency to maintain stable internal conditions despite external changes. Your body temperature remains near 37 degrees Celsius, whether the room is hot or cold. Your blood pH remains near 7.4, whether you eat acidic or alkaline foods. Your blood glucose remains in a narrow range despite irregular meals. These are not accidents. These are the products of elegant regulatory systems, feedback loops operating continuously, detecting deviation and initiating correction. The dopamine system has its own homeostasis, a baseline level of tonic activity, with the capacity for phasic bursts when genuinely important events occur. This is the natural state. Chronic overstimulation disrupted this balance. But now, in this period of reduced input, the regulatory mechanisms can reassert themselves. The neurons can adjust their receptor density. The synthesis and reuptake of dopamine can recalibrate. The sensitivity to natural rewards can return. This is not wishful thinking. This is documented neurobiology. Studies using PET scans, positron emission tomography, can visualize dopamine receptor density in living brains. When individuals reduce their exposure to supernormal stimuli, when they practice what we might call neural silence, the scans show measurable increases in receptor availability. The timeline varies, depending on the duration and intensity of prior overstimulation. But typically, significant upregulation begins within 48 to 72 hours. More substantial restoration occurs over weeks and months. The brain is patient. It will heal at its own pace if given the conditions it needs. Those conditions are simple, though not always easy. Reduce novel input. Allow boredom. Sit with discomfort. These phrases sound ascetic, like punishment, but they are actually descriptions of neuroplasticity. Boredom is not emptiness. Boredom is the state where the brain, absent external stimulation, begins to generate its own patterns. The default mode network activates, allowing for memory consolidation, autobiographical reflection, creative synthesis. The mind wanders, making unexpected connections, solving problems that conscious effort could not. This is why insights often arise in the shower, or during a walk, or just before sleep. Not because those activities are magical, but because they provide low-stimulation environments where the brain can engage its intrinsic processes. Tonight, you are creating such an environment. No screens, no scrolling, no rapid cuts between unrelated images. Just a single voice, speaking at a pace that allows for processing, for integration, for rest. Your alpha waves are deepening. The frequency is slowing toward the alpha-theta border, that liminal state between wakefulness and sleep. 8 cycles per second. 7, 6. The neurons in your cortex are firing in slow, synchronized waves, like wheat fields in gentle wind, bending together, rising together, in patterns that ripple across the surface. This is beautiful to observe on an EEG, electroencephalogram, the recording of brain electrical activity. Instead of chaotic spikes, instead of rapid, irregular patterns, the waves become smooth, regular, hypnotic, and as the waves slow, consciousness changes. The boundary between self and environment, usually so distinct, begins to soften. You are aware of your body, but not in the sharp, focused way of waking attention. More diffuse, more peaceful, like awareness spreading out into space. This is not loss of consciousness. This is a different kind of consciousness. One that humans experienced regularly before artificial lighting, before screens, before the acceleration of modern life. The consciousness of twilight, of firelight, of stories told in darkness. Your ancestors knew this state intimately, and your nervous system still remembers. The dopamine system continues its recalibration. With reduced input, the neurons in the ventral tegmental area are firing less frequently. This allows the postsynaptic neurons, the ones receiving the dopamine signal, to begin their adjustment. Inside these cells, genetic mechanisms are activating. Transcription factors are binding to DNA, initiating the production of new receptor proteins. This is upregulation at the molecular level. The genome responding to environmental conditions, generating the proteins needed for adaptation. It takes hours for mRNA to be transcribed, for proteins to be synthesized, for receptors to be transported to the cell membrane. But the process has begun. Your brain is healing itself while you rest. This is the wisdom of the biological system. It knows what to do. It has refined these mechanisms over millions of years of evolution. Your role is not to force the healing. Your role is simply to provide the conditions. And the primary condition is rest. As your brainwaves continue to slow, as your consciousness drifts toward the theta range, you may experience hypnagogic imagery, brief flashes of visual sensation, abstract patterns, fragments of scenes that make no logical sense. These are not dreams, not yet. These are the random activations of visual cortex, as the brain releases its grip on coherent narrative. The thalamus is reducing its filtering of sensory input. The cortex is beginning to operate more independently, generating its own activity. This is normal. This is the transition into sleep. Let the images come and go, without trying to understand them, without trying to hold them. They are like clouds passing across sky, interesting, transient, ultimately unimportant. Your breathing has changed. It has become slower, deeper, more regular. The respiratory control centers in your brainstem have shifted from conscious to automatic regulation. You no longer need to think about breathing. The medulla oblongata monitors carbon dioxide levels, adjusts respiratory rate, maintains oxygen homeostasis. This is one of the oldest regulatory systems in vertebrate biology, present in fish, in reptiles, in birds, in mammals. The brainstem, that most ancient part of your brain, taking care of what matters, keeping you alive, while the cortex, that most recent evolutionary development, finally gets to rest. Trust this process. Your body knows what to do. Your nervous system has been maintaining life, regulating systems, healing damage, since before you were born. The fetal brain develops its networks, establishes its connections, all without conscious input. The infant brain learns language, recognizes faces, builds models of the world, without deliberate instruction. Your adult brain maintains itself every night, clearing waste, consolidating memories, adjusting synaptic strengths. You do not need to supervise. You do not need to control. You simply need to allow. And in this allowing, in this surrender to the biological wisdom of rest, the dopamine system continues its restoration. The receptors are being manufactured. The sensitivity is being restored. The dynamic range is being rebuilt. Tomorrow, or the next day, or the day after, you will begin to notice. A conversation will feel more engaging. A meal will taste more vivid. A moment of sunlight will seem more beautiful. These are not psychological tricks. These are the experiences of a nervous system that has restored its capacity for genuine reward. But tonight, we are not seeking experiences. Tonight, we are seeking nothing. We are resting in the quiet space between stimulus and response, in the gap where healing happens, in the darkness where the brain rebuilds itself. Let the alpha waves deepen into theta. Let consciousness drift towards sleep. Let the body become heavy, warm, still. The restoration has begun, and it will continue through the night, through the deep delta frequencies of slow-wave sleep. But that is part 2 of this journey. For now, simply rest here, in alpha slowing toward theta, in the beginning of neural silence, in the first stage of dopamine restoration. You are descending now, not falling, but settling, like sediment through water, finding its natural level. The alpha waves that carried you through part 1 are giving way to something slower. Theta rhythms, 4 to 8 cycles per second, the frequency of deep meditation, of the hypnagogic state, of the boundary between waking and dreaming. Your conscious mind is still present, but its grip has loosened. And in this loosening, we can explore something deeper. The mechanism by which your brain protected itself, by becoming less sensitive. This is the biology of numbness. Let us return to the synapse, that microscopic gap between neurons, measuring approximately 20 to 40 nanometers across. A nanometer is 1/1000th of a meter. To give you perspective, a human hair is roughly 80,000 nanometers in diameter. The synaptic cleft is nearly invisible, yet it is where everything happens, where chemistry becomes information, where molecules become mind. On one side of this gap, the presynaptic terminal. This is the end of the axon, the long fiber extending from the neuron's cell body, specialized for sending signals. Inside this terminal, small spherical structures called vesicles. Each vesicle contains thousands of dopamine molecules, manufactured in the cell body, transported down the axon, packaged and ready for release. When an electrical signal, an action potential, arrives at the terminal, it triggers a cascade of events. Voltage-gated calcium channels open. Calcium ions rush into the cell. drawn by both electrical and concentration gradients. The calcium binds to sensor proteins on the vesicles. The vesicles fuse with the cell membrane. Their contents, the dopamine molecules, spill into the synaptic cleft. This is exocytosis, the process by which neurons communicate. The dopamine molecules, now free in the extracellular space, diffuse across the gap. Random molecular motion, Brownian movement, driven by thermal energy. Some molecules drift away, some are recaptured by transporters on the presynaptic membrane. But many reach the other side, the postsynaptic membrane, the receiving neuron. Here, embedded in the cell membrane, are the dopamine receptors. Specifically, we are concerned with two main types tonight. D1 receptors, which generally increase neuronal excitability, enhancing signal transmission, and D2 receptors, which typically decrease excitability, modulating and refining the signal. Both types are G-protein coupled receptors, meaning they do not directly open ion channels. Instead, when dopamine binds, the receptor changes shape, activating an intracellular G-protein. This G-protein initiates a cascade of biochemical events, a signaling pathway involving secondary messengers like cyclic AMP. The cascade amplifies the signal, allows for fine-tuned modulation, and ultimately affects the neuron's excitability and gene expression. This is not a simple on-off switch. This is a sophisticated analog system, capable of nuanced responses. Now imagine this system receiving constant input, hour after hour, day after day. The presynaptic terminals releasing dopamine in rapid succession. The receptors binding dopamine continuously. The intracellular cascades activated without pause. The postsynaptic neuron is in a state of chronic stimulation. This creates a problem at the molecular level. The receptors are proteins, complex three-dimensional structures folded with extraordinary precision. They exist in the cell membrane, that lipid bilayer separating the inside of the cell from the outside. Membranes are fluid, dynamic, constantly in motion. Proteins float within this membrane like icebergs in the ocean. But when receptors are continuously activated, when they are constantly bound by dopamine, the cell begins to perceive this as abnormal, as potentially dangerous. Why dangerous? Because overstimulation can lead to excitotoxicity. When neurons are driven too hard, when calcium levels rise too high, when metabolic demands exceed supply, cells can be damaged, synapses can degrade. In extreme cases, neurons can die. The cell has protective mechanisms against this. One of the most important is receptor downregulation. The process begins with phosphorylation. Kinase enzymes, activated by the continuous signaling, add phosphate groups to the intracellular tail of the dopamine receptor. This phosphorylation serves as a molecular tag, a signal that says, this receptor has been active too long. The phosphorylated receptor now attracts other proteins, arrestins, so named because they arrest receptor signaling. Beta-arrestin binds to the phosphorylated receptor, physically blocking it from interacting with G proteins. The receptor can still bind dopamine, but it can no longer transmit the signal intracellularly. It has been desensitized. But the cell does not stop there. The arrestin-bound receptor is now targeted for internalization. The cell membrane, That fluid lipid bilayer begins to dimple inward around the receptor. Clathrin proteins assemble on the inner surface of the membrane, forming a cage-like structure. This clathrin-coated pit deepens, narrows at the neck, and finally pinches off. The receptor, along with its patch of membrane, is now inside the cell. Enclosed in a clathrin-coated vesicle, the receptor has been removed from the cell surface. It can no longer respond to dopamine in the synaptic cleft. This is endocytosis, the opposite of exocytosis, the cell swallowing its own membrane, pulling receptors out of service. This happens continuously in your brain. Receptors are internalized, trafficked through the cell, either recycled back to the surface or degraded in lysosomes. It is a normal part of cellular maintenance. But when dopamine signaling is chronic, when stimulation is constant, the balance shifts. More receptors are internalized than are returned. The net result: fewer receptors on the cell surface. Downregulation. At the genetic level, changes are occurring as well. The continuous activation of dopamine signaling pathways affects gene expression. Transcription factors, proteins that bind to DNA and regulate which genes are active, respond to the signaling cascades. Some of these factors suppress the genes that code for dopamine receptors. Less mRNA is transcribed. Fewer receptor proteins are manufactured. The cell is not just removing existing receptors from the surface, it is reducing the production of new receptors. This is a deeper form of adaptation, one that takes longer to reverse. The time course matters here. Receptor internalization, the removal of receptors from the cell surface, can happen within minutes to hours. This is the acute response to overstimulation. Changes in gene expression, the reduction in receptor synthesis, develop over days to weeks. This is the chronic adaptation, and this is why recovery takes time. You cannot simply stop the stimulation for a few hours and expect full restoration. The genetic changes, the downregulated transcription, must reverse. New receptors must be manufactured, transported to the membrane, inserted into the lipid bilayer. This requires protein synthesis. It requires energy. It requires time for the cellular machinery to work. But here is what is crucial to understand. This process is not damage. This is adaptation. Your neurons have not been harmed by the overstimulation. They have responded intelligently to an abnormal environment. They reduced their sensitivity to protect themselves. And when the environment changes, when the constant input stops, they will adapt again. They will upregulate. They will restore sensitivity. This is neuroplasticity, the brain's capacity to modify its own structure and function in response to experience. You may have experienced this adaptation as emotional blunting, the sense that nothing feels quite satisfying, that activities which once brought pleasure now feel neutral, that you need more and more stimulation to feel less and less response. This is not depression, though it can resemble depression. This is receptor downregulation manifesting as subjective experience. The neurons in your nucleus accumbens, that structure involved in reward processing, have fewer available dopamine receptors. When you encounter something genuinely positive, when dopamine is released, there are fewer receptors to bind it. The signal is weaker, the experience is muted. The same thing happens with motivation. Dopamine in the prefrontal cortex is essential for executive function, for the ability to pursue goals that require sustained effort, for delayed gratification. When prefrontal dopamine receptors are downregulated, motivation becomes difficult, not because you lack willpower, but because the neurochemical substrate of motivation has been altered. The tasks that require effort, that do not provide immediate reward, feel almost impossible to initiate. You find yourself seeking the easy dopamine, the quick hit, the scroll that requires no effort. This reinforces the cycle, and the cycle extends to other neurotransmitter systems. Dopamine does not operate in isolation. It interacts with serotonin, with norepinephrine, with GABA, with glutamate. When one system becomes dysregulated, others compensate or become dysregulated themselves. Consider serotonin. This neurotransmitter, often associated with mood and well-being, has an inverse relationship with dopamine in some brain regions. When dopamine activity is chronically high, serotonin function can be suppressed. The RAFE nuclei, clusters of serotonergic neurons in the brainstem, may reduce their firing rate. Serotonin synthesis may decrease. Serotonin receptors may also downregulate. The result is a compounded deficit, not just reduced dopamine sensitivity, but reduced serotonergic tone as well. This is why the numbness feels so pervasive, why it affects not just motivation and pleasure, but mood and emotional regulation and sleep. Norepinephrine, the neurotransmitter of alertness and arousal, remains elevated. The locus coeruleus, the primary source of norepinephrine in the brain, has been firing constantly, responding to the perceived demands, the constant novelty, the continuous need for attention shifts. But chronic activation leads to receptor downregulation here as well. Specifically, alpha-2 autoreceptors on norepinephrine neurons. These receptors normally provide negative feedback, telling the neuron to reduce its firing rate when norepinephrine levels are sufficient. When these autoreceptors downregulate, the feedback is impaired. The neurons fire more, release more norepinephrine, but the system cannot regulate itself effectively. You feel wired, but tired, alert, but unable to focus. Your arousal system is stuck in the on position, even as you become desensitized to its signal. GABA, gamma-aminobutyric acid, the brain's primary inhibitory neurotransmitter, struggles to counterbalance this. GABA is released by interneurons, specialized cells that modulate the activity of excitatory neurons. When you feel calm, when anxiety subsides, when racing thoughts quiet, that is GABAergic inhibition at work. But chronic stress and overstimulation can impair GABAergic function. The synthesis of GABA may decrease. GABA receptors may become less sensitive. The result is reduced inhibitory tone. The excitatory signals, the glutamatergic drive, the constant firing, cannot be adequately dampened. Your mind races, your body remains tense, sleep becomes elusive. This is the full picture of downregulation. Not just one neurotransmitter, not just one receptor type, but a cascade of adaptations across multiple systems. Your brain has been trying to maintain equilibrium, to protect itself. But in the process, it has shifted to a new baseline. A baseline characterized by reduced sensitivity. impaired regulation, and chronic dysphoria. This feels terrible, but it is not permanent. Let us talk about what happens in the first 24 hours. When you stop the constant input, when you cease the scrolling and the switching and the rapid-fire stimulation, the immediate effect is withdrawal. Not withdrawal in the sense of physical dependence on a substance, but withdrawal in the sense of a system accustomed to constant input, suddenly receiving none. The dopamine neurons in the ventral tegmental area reduce their firing rate. They shift from phasic bursts, those rapid-fire activations in response to novelty, to tonic activity, the baseline firing that maintains readiness. The dopamine concentration in the synaptic cleft drops. The remaining receptors, those few that have not been internalized, detect this decrease, and the brain interprets this as a lack of reward, as boredom, as restlessness, as the urge to seek stimulation. This is uncomfortable. The discomfort is not imaginary, not psychological weakness. It is the subjective experience of neurons signaling unmet expectation. The nucleus accumbens, expecting its regular dopamine input, does not receive it. It sends signals to the prefrontal cortex, signals that manifest as urges, as impulses, as the compelling desire to reach for the device. The anterior cingulate cortex, that conflict monitor, detects the discrepancy between the goal of resting and the desire to seek stimulation. It generates the psychological tension, the feeling of struggle. This can manifest as irritability, as difficulty concentrating, as physical restlessness. You might feel the need to move, to fidget, to do something, anything. This is the most critical window. The first 6 to 12 hours of reduced stimulation. If you return to the behavior, if you pick up the device and begin scrolling, the dopamine system receives reinforcement. The neurons fire in phasic bursts again. Dopamine floods the depleted synapses. The few remaining receptors are activated, and the brain learns. The discomfort can be relieved by seeking stimulation. This strengthens the habit loop, makes the pattern more entrenched, makes future attempts at restoration more difficult. But if you can remain in the discomfort, if you can observe the urges without acting on them, something different happens. The dopamine neurons continue their baseline firing. No phasic bursts, no surges, just the steady, low-level activity that maintains the system without overstimulating it. The postsynaptic neurons, those cells with the downregulated receptors, detect this change. They sense that dopamine levels are lower, more consistent, no longer spiking. And slowly, the adaptive mechanisms begin to reverse. The process starts with the receptors that were internalized most recently. They are still inside the cell, enclosed in vesicles, but they have not yet been degraded. The cell can recycle them. When the continuous activation stops, when the phosphorylation and arrestin binding that triggered internalization is no longer occurring at high rates, the balance shifts. Some of the internalized receptors are trafficked back to the cell surface. They are inserted into the membrane. They become available to bind dopamine again. This begins within hours. It is not dramatic, not a sudden restoration, but a gradual increase in receptor availability. By 24 hours, measurable changes have begun. Functional imaging studies, using techniques like PET scans with radioactive tracers that bind to dopamine receptors, show slight increases in receptor availability. The magnitude depends on many factors: the severity of prior downregulation, individual genetic variations in receptor genes, sleep quality during the recovery period, nutritional status, particularly the availability of amino acid precursors for neurotransmitter synthesis. But the trend is consistent. Given reduced stimulation, the brain begins to upregulate. The subjective experience in this first 24 hours is variable. Some people report increased anxiety. The nervous system, no longer distracted by constant input, must confront whatever has been avoided. Worries about work, relationships, health, the future. These thoughts were always present, but they were drowned out by the noise of stimulation. Now, in the relative quiet, they emerge. This is not a failure of the process. This is the mind, accustomed to external focus, learning to manage internal states again. The prefrontal cortex, learning to regulate the amygdala, to modulate fear responses, to contextualize worries. This capacity has been there all along, but it has been underutilized. Like a muscle that has atrophied from lack of use. It feels weak at first, but it will strengthen. Other people report profound fatigue. The nervous system, which has been running on stress hormones and constant activation, finally relaxes. And in that relaxation, the accumulated sleep debt becomes apparent. You realize how tired you actually are. Not the wired tiredness of overstimulation, but genuine physiological fatigue. This is good. This is the body finally admitting its needs. Allow the sleep to come. Deep, extended, restorative sleep. This is when the most significant neural repair occurs. During sleep, particularly during slow-wave sleep, the brain undergoes metabolic restoration. The glymphatic system activates. This is the brain's waste clearance system, discovered only in 2012. During waking hours, the spaces between neurons are relatively small. But during deep sleep, brain cells actually shrink slightly, increasing the interstitial space by up to 60%. Cerebrospinal fluid flows through these enlarged spaces, washing away metabolic waste products. Beta amyloid proteins, tau proteins, lactate, all the byproducts of neural activity are flushed into the venous system and eventually cleared from the body. This clearance is essential for brain health. Without it, toxic proteins accumulate, cognitive function declines, neurodegenerative processes accelerate. Sleep is not optional. It is not a luxury. It is a biological necessity for neural restoration. And when you have been operating in a state of chronic overstimulation, your sleep debt is likely substantial. The brain will demand its rest. Allow this. Do not fight it with caffeine, with stimulation, with the belief that you should be able to push through. The fatigue is intelligent. It is your nervous system insisting on the conditions it needs to heal. By 48 hours, the upregulation process has progressed significantly. The receptors that were internalized are being returned to the surface at a higher rate. But more importantly, genetic changes are beginning to occur. The transcription factors that suppress dopamine receptor gene expression are becoming less active. The genes themselves, those stretches of DNA that code for D1 and D2 receptors, are being transcribed more frequently. messenger RNA is produced. Ribosomes translate this mRNA into receptor proteins. The proteins are folded, modified, transported to the cell membrane. New receptors appear on the surface of neurons. Receptors that were never internalized, but simply never existed because production was suppressed. This is true upregulation, not just recycling the old, but manufacturing the new. The time course for this process is days to weeks. Protein synthesis is not instantaneous. Each receptor protein must be constructed from amino acids, one by one, in the correct sequence. Over 400 amino acids for a typical dopamine receptor. Then it must be folded into the correct three-dimensional structure, a process assisted by chaperone proteins. Then it must be glycosylated, have sugar groups attached, which is necessary for proper function. Then it must be transported from the endoplasmic reticulum to the Golgi apparatus, to the cell membrane. Each step takes time, but each step is happening right now in millions of neurons throughout your brain. You will not feel this happening. There is no sensation associated with receptor upregulation, but you will notice the effects. Subtle at first, then more pronounced. A conversation that holds your attention when recently everything felt boring. A task that you can focus on when recently concentration was impossible. A meal that actually tastes good when recently food was just fuel. Music that moves you when recently it was just background noise. These are not placebo effects. These are the direct consequences of restored receptor density. Your neurons can once again detect and respond to dopamine signaling. The dynamic range has been restored. But the process is not linear. There will be difficult days. Days when the cravings return with intensity. Days when focus feels impossible. Days when everything seems gray and pointless. This is normal. This is the brain oscillating around a new equilibrium, rather than snapping directly to it. Think of it like a pendulum released from an extreme position. It swings back toward center, but it overshoots, swings the other way, gradually dampening, until it finally settles at rest. Your dopamine system is doing the same thing. Days of progress followed by days of difficulty. But the overall trend is toward restoration. The key is not to interpret the difficult days as failure. They are not evidence that the process is not working. They are evidence that your brain is a complex, dynamic system, not a simple machine. Healing is not monotonic. It involves fluctuation, adjustment, recalibration. The difficult days test your understanding. They ask the question, will you return to the old pattern, or will you trust the biology? And here is where we must talk about the concept of kindling. This is a term borrowed from neuroscience research on epilepsy, but it applies to any chronic brain state. Kindling refers to the phenomenon where repeated activation of a neural pathway makes that pathway easier to activate in the future. The neurons become sensitized, the synapses strengthen, the threshold for activation decreases. In epilepsy, this means that each seizure makes future seizures more likely. In addiction, this means that each relapse makes the addictive pattern more entrenched. And in dopamine dysregulation from overstimulation, this means that each return to the behavior, each time you give in to the urge and begin scrolling, strengthens that neural pathway, makes it easier to activate next time, makes recovery more difficult. This is not meant to induce shame. This is meant to convey biological reality. Your brain is learning constantly. It learns from repetition. When you perform an action, and that action is followed by dopamine release, the synapses involved in generating that action are strengthened through a process called long-term potentiation. The connection between seeing the device, picking it up, opening the app, and the dopamine surge becomes increasingly automatic. This is habit formation at the neural level, and habits, once formed, can be difficult to break. But they are not impossible to break. Habits can be overwritten through the same mechanisms of neuroplasticity that created them. When you choose not to act on the urge, when you experience the craving but do not reinforce it, you are weakening that pathway. The synapses, not receiving their expected reinforcement, gradually weaken through a process called long-term depression. The pathway becomes less automatic. The urge, though it may still arise, loses its compelling force. And simultaneously, you are strengthening alternative pathways. The pathway of noticing the urge without acting. The pathway of choosing rest over stimulation. The pathway of tolerating discomfort without immediately seeking relief. These become the new habits, the new automatic responses. This is why consistency matters more than perfection. If you maintain reduced stimulation most of the time, but occasionally have setbacks, the overall trajectory remains positive. The upregulation continues. The receptor density increases. The sensitivity returns. But if you oscillate, days of abstinence followed by binges of overstimulation, the system never stabilizes. You are kindling the old pathways even as you try to weaken them. The brain receives mixed signals. Recovery becomes prolonged, uncertain. So the guidance, the biological imperative, is this. Maintain the conditions for restoration. Allow the boredom. Sit with the discomfort. Do not seek the quick dopamine hit when the urge arises. Understand that the urge is not you. It is brain activity. It is neurons firing according to old patterns, and patterns can change. As you continue to rest, as your theta waves deepen, the cellular machinery of restoration continues its work. Ribosomes translating mRNA into receptor proteins, chaperones folding those proteins into functional shapes, transport vesicles carrying the receptors to the membrane, insertion of receptors into the lipid bilayer. All of this happening in darkness, in silence, without your conscious awareness or effort. This is the intelligence of the biological system. It knows what to do. It has refined these processes over billions of years of evolution. Your cells carry the wisdom of every ancestor who survived, who reproduced, who passed their genes forward. And that wisdom includes the capacity to restore equilibrium after disruption. Your role is simple. Provide the conditions. Reduce the input. Allow the rest. Trust the biology. The downregulation that occurred in response to overstimulation was not damage, was not failure, was not weakness. It was intelligent adaptation to an abnormal environment. And now, in response to reduced stimulation, in response to rest, the opposite adaptation is occurring. Upregulation. the restoration of sensitivity, the return of dynamic range. This is part 2 of the journey, the understanding of how numbness developed and how it will resolve. In part 3, we will explore the upregulation process in greater detail, the molecular mechanisms of recovery, the timeline of restoration, the subjective experiences as sensitivity returns. But for now, rest in this understanding. The downregulation was protective. The upregulation is inevitable, given the right conditions. Your brain wants to heal, and it is healing now. With every moment of rest, with every refusal to return to overstimulation, with every breath taken in the theta state of deep relaxation. The receptors are returning. The sensitivity is being restored. The future you, days or weeks from now, will experience the world with renewed clarity. Will feel motivation without forcing. Will find pleasure in simple things. Because your neurons will once again have the capacity to respond. Let your consciousness drift deeper now. The theta waves slowing toward the delta border. 4 cycles per second. 3, 2. The boundary of sleep approaching. The cellular work continues, but you need not witness it. You need not supervise. The biology knows its task, and you, the awareness, can finally rest in the darkness between waking and sleep, in the quiet space where healing happens, in the trust that the system, ancient and reliable, will restore itself. You are in the deep place now, the place between thoughts, where consciousness becomes diffuse, permeable, like morning fog over water. The theta waves that carried you through part 2 are dissolving into delta, 0.5 to 4 cycles per second, the slowest frequency, the rhythm of deep sleep, of cellular repair, of the body tending to itself in darkness. Your awareness may fade, may flicker, may disappear entirely into sleep. This is not only acceptable, it is ideal. Because what we are about to explore, the biology of upregulation, the return of sensitivity, happens most profoundly when you are not conscious, when the cortex is quiet, when metabolic resources can be redirected from thinking to healing. Let us speak about the 72-hour window. This is not an arbitrary number. This is the time frame, observed in multiple neurobiological studies, during which significant receptor upregulation begins to manifest. 3 days, 72 hours, 1,128 minutes, 103,680 seconds. Each one, a moment in which your neurons are adjusting, adapting, restoring. By hour 72, if you have maintained reduced stimulation, several processes have converged. The receptors that were internalized acutely in response to immediate overstimulation have been recycled to the surface. The genetic suppression of receptor synthesis has begun to reverse. New receptor proteins have been manufactured, transported, and inserted into membranes. The net result, a measurable increase in dopamine receptor density. Studies using radioligand binding assays show increases of 15 to 30% in receptor availability after 72 hours of reduced stimulation. This varies by brain region. The nucleus accumbens shows rapid upregulation. The prefrontal cortex, slightly slower. The dorsal striatum, involved in habit formation, slower still. But all regions trend upward. What does this mean at the level of experience? It means the world begins to come back into focus. Colors that seemed muted appear more vivid. Conversations that felt tedious become genuinely interesting. Tasks that required enormous willpower begin to feel naturally engaging. This is not psychological. This is not positive thinking. This is the direct consequence of neurons regaining their ability to respond to dopamine signaling. Consider a simple example. A piece of fruit, an orange, perhaps. In a state of receptor downregulation, eating an orange provides minimal pleasure. You register the taste, you consume the calories, but there is no real satisfaction. The sweetness exists. The flavor compounds activate taste receptors. Signals travel through the gustatory cortex. But the reward system, the dopamine neurons that normally respond to natural pleasures, are firing into down-regulated receptors. The signal is attenuated. The experience feels neutral. But after 72 hours of upregulation, the same orange becomes different. You peel it, and the scent is suddenly noticeable. The essential oils, limonene and citral, activating olfactory receptors, sending signals to the olfactory bulb, which connects directly to the amygdala and hippocampus. Scent and emotion and memory intimately linked. You segment the orange. Notice the texture, the way light refracts through the translucent flesh. You place a segment in your mouth, and the burst of juice is actually pleasurable. The sweetness registers not just as data, but as reward. The dopamine neurons fire. The signal reaches receptors that are now available. The nucleus accumbens responds, and you experience something you had forgotten. Simple pleasure. The kind that does not require novelty or intensity or artificial enhancement, just an orange. This is what upregulation restores. The capacity for subtle reward. The ability to find satisfaction in ordinary experience. This is not about lowering your standards. This is about restoring your biological capacity to register goodness when it is present. The molecular mechanism underlying this restoration is elegant. Inside the nucleus of your neurons, DNA is constantly being transcribed. Not all genes at once, but specific genes, depending on cellular conditions. Transcription factors, proteins that bind to specific DNA sequences, determine which genes are expressed. During chronic overstimulation, transcription factors like CREB, cyclic AMP response element binding protein, were suppressed in their activity. CREB normally promotes the expression of dopamine receptor genes, but chronic high dopamine signaling, paradoxically, reduces CREB activation. This is part of the negative feedback loop. The cell saying, "We have too much signal." Reduce receptor production. When stimulation decreases, when dopamine signaling returns to baseline, CREB activity increases again. The transcription factor binds to the promoter regions of dopamine receptor genes. RNA polymerase is recruited. The gene is transcribed into messenger RNA. The mRNA exits the nucleus, travels to the cytoplasm, binds to ribosomes, and the ribosome, that molecular machine, reads the mRNA sequence 3 nucleotides at a time. Each triplet codes for a specific amino acid. Transfer RNA molecules bring the appropriate amino acids. The ribosome links them together, one by one, forming a growing chain. This is translation. The information in the gene, becoming the physical structure of a protein. For a dopamine D2 receptor, this process produces a chain of 443 amino acids. The chain spontaneously begins to fold, guided by the chemical properties of the amino acids. Hydrophobic amino acids cluster together, away from water. Hydrophilic amino acids face outward, toward the aqueous environment. Disulfide bonds form between cysteine residues, stabilizing the structure. The protein folds into 7 transmembrane alpha helices, the characteristic structure of G protein-coupled receptors. This folding is assisted by chaperone proteins, molecular helpers that prevent misfolding and aggregation. The process takes minutes. Once properly folded, the receptor undergoes post-translational modifications. Glycosylation, the addition of sugar molecules, occurs in the endoplasmic reticulum. These sugar groups are important for protein stability and function. The receptor is then packaged into transport vesicles. These vesicles bud off from the endoplasmic reticulum, travel through the cytoplasm, fuse with the Golgi apparatus. In the Golgi, further modifications occur. Additional sugar groups may be added or modified. The receptor is sorted, tagged with signals that direct it to the plasma membrane. Finally, it is packaged into vesicles that travel to the cell surface. The vesicle approaches the plasma membrane. Snare proteins on the vesicle and membrane interact, bringing the 2 lipid bilayers into close proximity. The membranes fuse. The vesicle opens to the extracellular space, and the receptor, that carefully constructed protein, is now embedded in the cell membrane. Its extracellular domain facing the synaptic cleft, ready to bind dopamine. Its intracellular domain ready to activate G proteins and initiate signaling cascades. A new receptor, functional and available. This is upregulation at the molecular level. And this is happening simultaneously in millions of neurons, not just in one brain region, but throughout the dopaminergic pathways. The ventral tegmental area, the substantia nigra, projecting to the nucleus accumbens, the prefrontal cortex, the dorsal striatum, the amygdala. Each neuron, each synapse, undergoing its own process of restoration. The collective effect, a brain-wide increase in dopamine sensitivity. But upregulation is not just about receptor density. It is also about receptor function. Even when receptors are present on the membrane, their efficiency can vary. During chronic overstimulation, receptors can become less responsive through a process called desensitization. The receptor is still there. It can still bind dopamine, but the downstream signaling is impaired. The G protein coupling is less efficient. The intracellular cascades are less robust. The signal-to-response ratio is degraded. Upregulation restores this efficiency. The receptors that remain on the membrane become more sensitive. The G proteins, heterotrimeric complexes of alpha, beta, and gamma subunits, are more readily activated. The production of cyclic AMP, that crucial second messenger, increases in response to receptor activation. Downstream targets like protein kinase A are activated more effectively. The entire signaling cascade becomes more responsive. This is functional upregulation. Not just more receptors, but more effective receptors. The timeline for full restoration varies. Acute receptor recycling, The return of internalized receptors to the surface occurs within hours to days. Genetic upregulation, increased synthesis of new receptors, becomes significant by 3 to 7 days. Full restoration of baseline receptor density, depending on the severity of prior downregulation, may take 2 to 4 weeks. Restoration of optimal function, including normalization of all related neurotransmitter systems, may take 6 to 12 weeks. These are averages. Individual variation is substantial. Factors that accelerate upregulation include sleep quality, deep sleep, specifically slow-wave sleep, is when the most robust neural restoration occurs. During stage 3 and stage 4 sleep, when delta waves dominate, growth hormone is released from the pituitary gland. Growth hormone stimulates protein synthesis throughout the body, including in the brain. It promotes the production of insulin-like growth factor 1, IGF-1, which supports neuronal health and plasticity. The more time you spend in deep sleep, the more resources are available for receptor synthesis and membrane remodeling. Nutrition also matters. Dopamine synthesis requires specific amino acid precursors, primarily tyrosine, an amino acid obtained from dietary protein. Tyrosine is converted to L-dopa by the enzyme tyrosine hydroxylase. L-dopa is converted to dopamine by aromatic L-amino acid decarboxylase. If tyrosine is insufficient, dopamine synthesis is limited. Similarly, the synthesis of serotonin requires tryptophan. The synthesis of norepinephrine requires dopamine as a precursor. Adequate dietary protein, containing all essential amino acids, supports neurotransmitter production. Micronutrients play roles as well. Iron is a cofactor for tyrosine hydroxylase, the rate-limiting enzyme in dopamine synthesis. Vitamin B6, pyridoxal phosphate, is a cofactor for aromatic L-amino acid decarboxylase. Folate and vitamin B12 are involved in 1-carbon metabolism, supporting the synthesis of neurotransmitter precursors. Magnesium modulates NMDA receptors and supports neuronal excitability. Zinc is involved in neurotransmitter packaging and release. These are not magic bullets, but deficiencies, even marginal deficiencies, can impair the restoration process. Physical movement supports upregulation through multiple mechanisms. Exercise increases blood flow to the brain, delivering oxygen and nutrients. It stimulates the production of brain-derived neurotrophic factor, BDNF, a protein that supports neuronal survival and synaptic plasticity. BDNF enhances long-term potentiation, the strengthening of synapses, which is essential for learning and habit formation. It also promotes neurogenesis, the birth of new neurons, particularly in the hippocampus. Moderate exercise, not intense enough to cause stress, but sufficient to elevate heart rate, appears optimal. Walking, swimming, cycling, yoga, activities that combine movement with presence, with attention to the body. But perhaps most important is the absence of re-stimulation. Every time you return to the supernormal stimulus, every time you pick up the device and begin scrolling, you interrupt the upregulation process. The dopamine surge, even brief, even moderate, signals to the neurons that high stimulation is still occurring. The transcription factors that were beginning to promote receptor synthesis are inhibited again. The receptors that were being manufactured may not be inserted into the membrane. The system hesitates, uncertain whether to continue adapting toward lower sensitivity or higher sensitivity. Recovery stalls. This is why the 72-hour window is emphasized. It is not that nothing happens after 72 hours if you relapse. It is that the first 72 hours are when the most vulnerable aspects of recovery occur, when the acute withdrawal is most intense, when the temptation to return to old patterns is strongest, when the biological momentum toward upregulation is still building. If you can maintain reduced stimulation through this period, the process becomes self-reinforcing. The increased receptor density makes natural rewards more satisfying. The improved sleep makes emotional regulation easier. The reduced cravings make continued abstinence less effortful. A positive feedback loop emerges. But if you relapse during this window, the momentum is lost. The brain receives the message that the old pattern is still active. The dopamine system does not fully commit to upregulation. You remain in a state of partial recovery, neither fully downregulated nor fully restored. This is the most uncomfortable state. Enough sensitivity to feel cravings intensely, but not enough to feel satisfaction from natural rewards. So the guidance is clear. Commit to the 72 hours. 3 days of minimal digital stimulation. No scrolling through feeds. No rapid switching between apps. No content designed for maximum engagement. This does not mean complete isolation. You can read books, but physical books, at your own pace. You can have conversations, but in person, without multitasking. You can listen to music, but intentionally, allowing full attention. You can walk, observe, think. You can be bored. Boredom is not emptiness. Boredom is the state where the mind, absent external input, begins to generate its own activity. And that activity is valuable. The default mode network, active during rest, supports autobiographical memory, future planning, theory of mind. It allows you to consolidate experiences into coherent narratives, to imagine possibilities, to understand yourself and others. This network has been suppressed during chronic overstimulation. The constant external focus, the endless stream of content, prevents the inward turn. But in boredom, in rest, the default mode network activates, and you reconnect with aspects of yourself that were obscured. By day 4 or 5, if the process has been maintained, you will notice shifts. Motivation begins to return. Not the forced motivation of willpower, but the natural arising of interest. A project that seemed impossible to start suddenly feels manageable. A conversation you were dreading becomes something you look forward to. A creative idea emerges, fully formed, from nowhere apparent. These are signs of prefrontal dopamine function restoring. The executive networks, capable once again of maintaining goal representations, of delaying gratification, of executing complex plans. The emotional tone also shifts. Where there was flatness, subtle variations in mood return. Where there was anxiety, moments of calm appear. Where there was irritability, patience emerges. This reflects the normalization of serotonergic function. The raphe nuclei, those clusters of serotonin neurons, have had time to replenish their stores. Serotonin receptors, which also downregulated during chronic stress, have begun to upregulate. The modulation of mood and emotional reactivity improves. Sleep improves as well. The difficulty falling asleep, the racing mind, the restless body, these diminish. You lie down, close your eyes, and within a reasonable time, sleep comes. Not the collapse of exhaustion, but the natural transition from waking to sleeping. And the sleep itself becomes more restorative. More time in slow-wave sleep. More time in REM, rapid eye movement sleep, when dreams occur. Both stages are essential. Slow-wave sleep for physical restoration and memory consolidation. REM sleep for emotional processing and creative synthesis. A balanced sleep architecture indicates a nervous system returning to equilibrium. By week 2, the changes become more pronounced. Activities that once seemed pointless begin to hold meaning. A walk in nature is not just exercise, it is genuinely pleasant. A conversation with a friend is not just social obligation, it is nourishing. A meal is not just fuel, it is an experience. This is the return of dynamic range, the ability to experience gradations of reward, from subtle to intense. Not everything needs to be maximally stimulating to be worthwhile. In fact, the maximally stimulating becomes less appealing. The frantic pace, the constant novelty, the artificial urgency feels exhausting now. You begin to prefer slower, deeper, more sustained engagement. This preference shift reflects changes in neural circuitry. The habit loops, those automatic pathways linking cue to craving to response to reward, are being rewritten. The old pathway, see phone, feel urge, pick up phone, scroll, receive dopamine, is weakening. Each time you experience the cue and do not follow the habitual response, the synapses in that pathway undergo long-term depression. The connection weakens, the automaticity fades, and new pathways strengthen. See boredom, tolerate discomfort, engage with present moment, experience subtle reward. This becomes the new automatic response, not through force, but through repetition, through the natural plasticity of the nervous system. Neuroplasticity is often discussed as if it only applies to learning skills or recovering from injury. But it applies to everything the brain does. Every habit, every preference, every emotional pattern is encoded in synaptic strengths, and synaptic strengths can change. The mechanisms are well understood. Long-term potentiation, the strengthening of frequently used synapses through increased AMPA receptor insertion and dendritic spine growth. Long-term depression, the weakening of infrequently used synapses through AMPA receptor removal and spine shrinkage. Synaptic pruning, the elimination of weak or unused connections to increase efficiency. These processes occur constantly, throughout life, in response to experience. What we call personality, temperament, character, are to a significant extent patterns of synaptic connectivity. Stable patterns, yes, but not immutable. The circuits that make you reach for distraction when uncomfortable can be weakened. The circuits that allow you to sit with discomfort can be strengthened. This is not self-improvement through positive thinking. This is neural rewiring through behavior change. By week 3 or 4, assuming consistency, you may notice something remarkable. You do not miss the constant stimulation. The thought of scrolling through feeds for an hour feels not tempting, but vaguely repulsive, like the thought of eating candy for breakfast. Technically possible, but clearly not what you want. This is not willpower. This is preference change arising from restored dopamine sensitivity. Your brain now finds greater reward in real-world engagement than in artificial stimulation. The natural hierarchy of rewards has been restored. Social connection, genuine conversation, eye contact and tone of voice and shared presence activates reward circuits more effectively than text messages. Physical movement. The proprioceptive feedback of muscles working, the rhythm of breath, the feeling of competence becomes intrinsically rewarding. Creative work, the flow state of deep focus, the satisfaction of solving problems, the tangible output, provides sustained dopamine release. Learning, encountering new ideas, integrating them with existing knowledge, experiencing the aha moment of understanding is deeply rewarding. These are what dopamine was designed for, not the hyper-novelty of infinite scroll, but the genuine rewards of a life fully lived. And yet, vigilance remains necessary. The old pathways, though weakened, are not eliminated. They exist as latent potential, ready to reactivate if the behavior returns. This is why individuals recovering from addiction speak of being in recovery rather than recovered. The vulnerability persists. One use can reactivate dormant circuits. One binge can trigger the cascade back toward downregulation. This is not a moral judgment. This is the reality of neuroplasticity. Pathways that were deeply ingrained through months or years of repetition do not disappear completely. They fade, they weaken, but they remain. The practical implication is clear. Maintain boundaries. Structure your environment to support your recovery. Remove temptation where possible. Delete apps that triggered overstimulation. Use website blockers during vulnerable hours. Establish phone-free zones and times. Replace the old habits with new ones. When you would have scrolled, read instead. When you would have checked notifications, take 3 conscious breaths instead. When you would have sought digital distraction, go outside instead. These replacements are not arbitrary. They are the new pathways you are strengthening, the new habits forming through repetition. And celebrate the small victories. The morning you wake without immediately reaching for your phone. The evening you spend in conversation without interruption. The hour you spend in focused work without checking email. These are not trivial. These are evidence of neural rewiring, of synapses strengthening in the pathways you choose, of a brain adapting toward the patterns you reinforce. This is neuroplasticity in action. This is you, shaping your own neural architecture through deliberate choice. The upregulation process. This restoration of sensitivity is both biological and behavioral. The receptors will increase in density through the mechanisms we have described. But the habits, the automatic patterns, require conscious participation. You cannot passively wait for your brain to heal. You must actively create the conditions for healing. And then, within those conditions, the biology will do what it knows how to do. Adapt, restore, find equilibrium. As we move toward part 4, as your consciousness drifts deeper into delta, into the realm of dreamless sleep, let this understanding settle. Upregulation is not instant. It is a process measured in days and weeks, but it is inevitable, given consistency. Your neurons are designed to restore sensitivity after overstimulation. Evolution has refined these mechanisms over millions of years. Trust them. Support them with your choices, and they will return you to a state of balance. The deep delta waves now. 1 cycle per second. The slowest rhythm, the frequency of complete rest. Your body, heavy and still. Your breath, slow and regular. Your mind, quiet. The upregulation continues in the darkness. Receptors being synthesized, transported, inserted. Synapses being pruned, strengthened, optimized. Networks being rewired toward new patterns. All of this happening without effort. The biology, ancient and wise, tending to itself. In Part 4, we will explore homeostasis, the final stage of restoration, when all systems return to balanced baseline, when the nervous system finds its sustainable rhythm, when dopamine and serotonin and norepinephrine and GABA all operate in coordinated harmony. But that is later. For now, rest in the upregulation, in the knowledge that every hour of reduced stimulation, every night of deep sleep, every choice to remain present rather than seek distraction is strengthening your recovery. The receptors are returning. The sensitivity is being restored. The capacity for genuine reward, for natural motivation, for sustainable pleasure is coming back. Trust the process. Trust the biology. Sleep now. You have descended into the deepest place, the delta frequency, where consciousness dissolves, where the self, that construct of narrative and memory, temporarily ceases, where only the body remains, breathing, restoring, existing without observer. This is the realm of homeostasis, the biological tendency toward balance, the invisible intelligence that maintains your internal universe while you sleep. Homeostasis is not a destination. It is a process, a continuous dynamic adjustment, maintaining stability through constant change. Your body temperature fluctuates throughout the day, yet remains near 37 degrees Celsius. Your blood pressure rises with activity and falls with rest, yet maintains an average that sustains perfusion. Your blood glucose spikes after meals and drops during fasting, yet remains within a range that prevents harm. These are not accidents. These are the products of regulatory systems, feedback loops operating at every level of biological organization, from molecules to cells to organs to the organism as a whole. The nervous system has its own homeostasis, neural homeostasis, the maintenance of stable patterns of activity despite changing inputs. When stimulation increases, neurons adapt by reducing sensitivity. When stimulation decreases, neurons adapt by increasing sensitivity. This is what we have been exploring. Downregulation in response to overstimulation. Upregulation in response to rest. The system seeking its equilibrium point. That equilibrium point, that baseline, is not arbitrary. It is calibrated by evolution, by millions of years of natural selection, by the survival and reproduction of organisms whose nervous systems could maintain stable function across varying environments. Your baseline dopamine sensitivity, the receptor density and signaling efficiency that feels normal to you, is the product of your genetic inheritance and developmental history. It is your biological set point. chronic overstimulation pushed you far from that set point. Weeks or months of supernormal stimulation drove the system into a new state, a state of reduced sensitivity, elevated stress hormones, impaired regulation. This felt terrible because it was far from equilibrium. The system was expending enormous energy trying to compensate, to maintain function, despite being pushed to an extreme. But it could not return to baseline while the excessive stimulation continued. Now, after days or weeks of reduced input, the system is approaching its set point again. The receptors have upregulated. The neurotransmitter synthesis has normalized. The stress response has downregulated. The sleep architecture has restored. You are nearing homeostasis. What does homeostasis feel like? It feels like nothing remarkable. And that is the point. Homeostasis is the absence of dysregulation. It is not euphoria, not constant pleasure, not perpetual motivation. It is the capacity to experience the full range of human emotion and motivation in appropriate response to circumstances. Joy when there is genuine cause for joy. Sadness when there is loss. Motivation when there are meaningful goals. Rest when there is nothing demanding action. This may sound disappointing. In a culture addicted to peak experiences, to constant positivity, to the pursuit of optimal states. But homeostasis is what allows sustainable flourishing. The baseline from which you can respond, create, connect, grow, without burning out, without depleting yourself, without requiring constant external stimulation to feel okay. Let us explore what homeostasis looks like at different levels of organization. At the molecular level, neurotransmitter synthesis matches release and degradation. Dopamine production in the substantia nigra and ventral tegmental area occurs at a rate that maintains tonic activity without depletion. The enzymes involved, tyrosine hydroxylase and aromatic L-amino acid decarboxylase, operate at baseline efficiency. Vesicles are filled with dopamine at a rate that matches vesicle fusion and neurotransmitter release. The system is in steady state. Production equals consumption. No excess, no deficit. At the synaptic level, receptor density matches the typical dopamine concentration. Enough receptors to detect signal clearly. Not so many that the system is overly sensitive to minor fluctuations. The balance between excitation and inhibition is maintained. Glutamate, the primary excitatory neurotransmitter, activates neurons when appropriate. GABA, the primary inhibitory neurotransmitter, restrains activation when appropriate. The network oscillates around stable activity levels. Responsive but not hyperactive. Calm but not sluggish. At the circuit level, brain networks coordinate efficiently. The default mode network activates during rest, supporting introspection and memory consolidation. The salience network detects important stimuli and directs attention appropriately. The executive control network maintains goals and regulates behavior. These networks interact fluidly, without one dominating or suppressing the others excessively. Attention can shift between internal and external focus, between focused work and creative wandering, between social engagement and solitary reflection. At the systems level, the autonomic nervous system maintains balance. The sympathetic branch, responsible for arousal and stress response, activates during challenge, The parasympathetic branch, responsible for rest and digestion, activates during safety. In homeostasis, neither branch dominates chronically. Heart rate variability, the beat-to-beat variation in heart rate, is high. This indicates flexibility, the capacity to shift between arousal and calm as needed. Low heart rate variability indicates a system stuck in one state, usually sympathetic dominance, chronic stress arousal. High variability indicates health, adaptability, resilience. The hypothalamic pituitary adrenal axis, the HPA axis, also normalizes. This is the body's primary stress response system. When threat is detected, the hypothalamus releases corticotropin-releasing hormone. This triggers the pituitary to release adrenocorticotropic hormone. This triggers the adrenal glands to release cortisol. Cortisol mobilizes energy, suppresses non-essential functions, prepares the body for action. This is adaptive for acute stress, but chronic activation, chronic elevated cortisol, is harmful. It impairs immune function, disrupts sleep, damages the hippocampus, contributes to mood disorders. In homeostasis, the HPA axis responds appropriately to stress and then returns to baseline. Cortisol rises in the morning to promote wakefulness. It declines through the day. It reaches its lowest point in the evening, allowing melatonin to rise and sleep to occur. When stress arises, cortisol increases, but not excessively. When the stressor resolves, cortisol returns to baseline within hours. The system is responsive but regulated. Sleep becomes the most reliable indicator of homeostasis. When the nervous system is balanced, sleep occurs naturally. You feel sleepy at an appropriate time in the evening. You fall asleep within 10 to 20 minutes of lying down. You progress through the sleep stages in normal proportion. Approximately 75 to 80% non-REM sleep, including deep, slow-wave sleep in the first half of the night. Approximately 20 to 25% REM sleep, increasing in duration toward morning. You wake feeling restored, not perfect, not euphoric, but rested. Ready for the day without requiring artificial stimulation to feel functional. During deep sleep, the brain performs its most profound maintenance. The glymphatic system, most active during slow-wave sleep, clears metabolic waste at 10 to 20 times the waking rate. Cerebrospinal fluid flows through the expanded interstitial spaces. Beta amyloid, tau protein, lactate. All the byproducts of neural activity are flushed away. This clearance is essential. Without it, these proteins accumulate, aggregate, interfere with neural function. Over years and decades, this accumulation contributes to cognitive decline and neurodegeneration. But nightly, if sleep is adequate, the system clears itself. Slow-wave sleep is also when synaptic homeostasis occurs. During waking, synapses strengthen in response to experience. Every input, every thought, every action potentiates some connections. This is learning, but unchecked strengthening would lead to saturation. All synapses maximally strengthened. No room for new learning. Excessive energy consumption. So during slow-wave sleep, synaptic downscaling occurs. Weaker synapses, those not reinforced by repeated activation, are pruned. Stronger synapses, those representing important information, are preserved. The signal-to-noise ratio improves. Memory consolidates while unimportant details fade. REM sleep serves different functions. During REM, the brain is highly active, almost as active as during waking, but the activation is not driven by external input. It is generated internally by the brainstem, creating the vivid imagery and narrative of dreams. The exact purpose of REM sleep remains debated, but evidence suggests it is crucial for emotional processing, for integrating emotional experiences, for rehearsing responses to threats in a safe context, for creative problem-solving through novel combinations of memories. Dreams can be bizarre, illogical, disconnected. This is because the dorsolateral prefrontal cortex, responsible for logical reasoning and reality testing, is largely inactive during REM. The associations form freely, without executive constraint. This allows for creative synthesis, for unexpected connections. Many scientific discoveries, artistic creations, and personal insights have emerged from dreams. Not because dreams are mystical, but because they represent unconstrained neural activity, exploring the space of possible connections. In homeostasis, you dream. Not always remembered, but dreams occur in every REM period. 4 to 6 REM periods per night in a normal sleep architecture. The dreams may be mundane or elaborate. They may reflect daily concerns or venture into surreal territory. But their occurrence indicates that the brain is cycling through its natural stages, that REM mechanisms are functioning, that emotional processing is occurring. Upon waking, in a state of homeostasis, you do not immediately seek stimulation. You do not reach for the phone before opening your eyes. You lie for a moment, aware of the transition from sleep to waking. You notice your body, your breath, the quality of light. You rise when ready. You move through morning routines with presence rather than urgency. There is no frantic need to check what happened while you slept. No anxiety that you missed something important. Just the morning. Throughout the day, attention is flexible. You can focus deeply when a task requires it. The prefrontal cortex maintains goal representations without excessive effort. Distractions arise, but they do not capture attention involuntarily. You notice them, assess relevance, and either engage or return to the task. This is executive control functioning normally. Not perfectly, because no human has perfect attention, but adequately, sufficient for productive work and meaningful engagement. When work is complete, you can transition to rest. You do not need to force yourself to stop. You do not feel compelled to keep checking, keep working, keep doing. The task is done, and that is sufficient. You shift to leisure without guilt. You read, or walk, or converse, or simply sit. And these activities are genuinely satisfying. Not thrilling, not euphoric, but pleasant. Enough. Social interactions feel natural. Conversations flow without excessive self-consciousness. You listen to others without planning your next statement while they speak. You share your thoughts without rehearsing them internally first. Eye contact is comfortable rather than awkward or avoided. The subtle nonverbal cues, facial expressions, and tone and body language are processed without conscious effort. This is social cognition functioning normally. The mirror neuron system, the theory of mind networks, operating below awareness, allowing genuine connection rather than performative interaction. Emotions arise in proportion to circumstances. Good news brings pleasure. Setbacks bring disappointment. Conflict brings frustration. But the emotions are manageable. They do not overwhelm. They do not spiral into catastrophic thinking. The prefrontal cortex maintains regulation over the amygdala. The ventromedial prefrontal cortex, in particular, provides top-down inhibition of fear responses. You feel the emotion, you acknowledge it, and it passes, leaving space for the next moment. This emotional flexibility is one of the clearest signs of homeostasis. When the nervous system is dysregulated, emotions are either blunted or overwhelming. Either you feel nothing, numbness, disconnection, or you feel everything too intensely, reactivity, volatility. Both extremes indicate imbalance. Homeostasis is the capacity to feel appropriately, to have emotions that match the situation in intensity and duration, to recover from negative emotions within hours rather than days, to sustain positive emotions without needing constant reinforcement. The dopamine system, in homeostasis, operates with dynamic range. Small rewards produce small responses. Large rewards produce large responses. The system is calibrated. A kind word from a stranger brings a brief lift in mood. A significant achievement brings sustained satisfaction. A meaningful relationship brings deep fulfillment. The gradient from subtle to profound is preserved. And importantly, the absence of reward does not feel intolerable. Neutral moments are neutral, not unpleasant, not something to escape, just the baseline, the space between events. This is the capacity that chronic overstimulation destroys. The ability to tolerate neutrality, when every moment without stimulation feels like deprivation, when baseline feels like depression. You are trapped in a hedonic cycle, seeking more input to feel okay. But in homeostasis, neutral is acceptable. You can sit in a waiting room without needing to check your phone. You can stand in line without feeling impatient. You can lie in bed before sleep without needing entertainment. The absence of stimulation is not threatening. It is just silence. And in that silence, the mind can wander, can reflect, can rest. This capacity for rest is perhaps the most valuable aspect of homeostasis. The ability to do nothing and be okay, to exist without constantly producing or consuming or achieving, to simply be. This is not laziness. This is the foundation of sustainable engagement. You cannot work effectively if you cannot also rest effectively. You cannot create from a depleted reservoir. You cannot connect authentically if you are always performing. Rest is not the absence of productivity. Rest is the source of productivity. In homeostasis, rest comes naturally. When you are tired, you feel tired. Not wired and tired, not restless exhaustion, just tired. And you can respond to that signal. You can lie down, close your eyes, and allow sleep to come. Without forcing, without frustration, without requiring elaborate sleep hygiene protocols. The body knows what to do. The circadian rhythm is aligned. Adenosine, that molecule that accumulates during waking and promotes sleep, reaches sufficient levels. Melatonin rises in response to darkness. Core body temperature drops slightly. All the conditions for sleep are present. And consciousness releases its grip. The transition from waking to sleep in homeostasis is smooth, not abrupt, not jarring. a gentle descent through the brainwave frequencies, beta to alpha to theta to delta, each stage flowing naturally into the next. Awareness dimming, diffusing, dissolving, until only the breath remains. And then, even the breath fades from awareness, and you are asleep. Sleep itself, in homeostasis, is deep and undisturbed. You may wake briefly once or twice. This is normal, but you return to sleep easily. The night passes, the sleep stages cycle, the cellular restoration occurs, the glymphatic clearance continues, the synaptic pruning proceeds, the memory consolidation completes, and in the morning, you wake naturally, not to an alarm, though you may use one as backup, but to the internal signal that sleep is complete. This is what homeostasis offers. Not transcendence, not constant bliss, not superhuman performance, but ordinary human functioning. The capacity to work and rest, to feel and regulate, to connect and reflect, to engage with life as it is, rather than constantly seeking escape or enhancement. And this ordinary functioning is extraordinary in its value, because it is sustainable, because it does not require constant effort to maintain, because it allows you to live without being at war with your own biology. The nervous system, in balance, supports you, rather than fighting against you. Maintaining homeostasis, once achieved, requires awareness, not vigilance, not anxiety, but awareness. Awareness of your patterns. Awareness of early signs of dysregulation. Awareness of the environmental and behavioral factors that push you toward imbalance. You will notice, perhaps, when you begin to feel restless, when neutral moments start to feel uncomfortable again, when you reach for distraction more frequently. These are early signals. They indicate that the dopamine system is beginning to adapt to increased stimulation again, that you are approaching the edge of your sustainable range. The response is simple. Reduce input, increase rest. Return to the behaviors that support homeostasis, not as punishment, not with shame, but with the same matter-of-fact approach you would take to any biological need. If you were thirsty, you would drink. If your nervous system is overstimulated, you reduce stimulation. It is that straightforward. Over time, this becomes intuitive. You develop a felt sense of your own regulation. You can detect subtle shifts in your baseline. You can intervene early, before dysregulation becomes entrenched. This is not obsessive self-monitoring. This is attunement. The same way you know when you are hungry or tired or need to move. You know when your nervous system needs rest, and you can provide that rest, not just through sleep, but through daily choices. Moments of doing nothing, walks without podcasts, meals without screens, conversations without multitasking. These are not deprivations. These are gifts to your nervous system. Spaces of low input where restoration can occur continuously, preventing the accumulation of dysregulation. Maintaining homeostasis through consistency rather than requiring periodic dramatic interventions. This is the sustainable path, not oscillating between overstimulation and forced detox, but living in a way that respects your neurobiology, that acknowledges you are a biological organism with specific needs, that provides those needs daily, as naturally as breathing. The nervous system, given these conditions, will maintain itself. It will repair minor perturbations. It will adapt to changing demands. It will support your engagement with life without requiring you to constantly manage or override it. This is the promise of homeostasis. And so we arrive at the end of this journey. You began in overstimulation, in high beta, in dysregulation. We descended through understanding. The biology of downregulation, why numbness developed. The process of upregulation, how sensitivity returns. And now, homeostasis, the balanced baseline that allows sustainable flourishing. Your brain has been working throughout this narration. The slow rhythm of these words, the absence of rapid cuts and novelty, the permission to rest, to drift, to allow consciousness to dissolve. All of this has supported your nervous system's movement toward balance. The alpha waves, the theta waves, the delta waves, each frequency a step deeper into restoration. Whether you are still awake, drifting, or deeply asleep, the biology continues its work. Receptors being synthesized and inserted, neurotransmitters being produced and released in balanced proportions, networks being pruned and strengthened according to use, the glymphatic system clearing waste, the immune system supporting neural health, all the invisible processes of homeostasis, occurring without effort, without supervision, without your conscious awareness required. Trust this. Trust the ancient wisdom of your biology. Trust that the system wants to heal, wants to find balance, wants to support your flourishing. Your role is simply to provide the conditions. Rest, nutrition, movement, social connection, meaningful engagement, and reduced artificial stimulation. The conditions under which human nervous systems evolved to function. When you wake, whether in minutes or hours, you will not be suddenly transformed. You will not feel completely different. But you will have taken steps toward restoration. And if you continue, if you maintain the practices that support homeostasis, over days and weeks, the changes will compound. The sensitivity will return. The motivation will arise naturally. The pleasure will be found in simple things. And you will remember what it feels like to be okay. Not chasing, not escaping, not constantly seeking, just okay. This is the gift of homeostasis. This is what your nervous system offers when given the chance. Balance, resilience, the capacity for genuine engagement with life. Sleep now. Rest deeply. Let the delta waves carry you into complete restoration. Let the cellular machinery continue its work. Let the dopamine receptors return to the surface of your neurons. Let the synapses prune and strengthen according to your choices. Let the glymphatic system clear what needs clearing. Let the circadian rhythm align. Let the stress response downregulate. Let the parasympathetic nervous system restore your body. And when you wake, begin again. With awareness, with intention, with trust in your biology, creating the conditions for homeostasis, living in a way that respects your neurobiology, allowing the nervous system to do what it knows how to do. Maintain balance, find your sustainable rhythm, and flourish in the ordinary, extraordinary miracle of being human. With a nervous system in homeostasis, supporting your life, rather than struggling against it. This is the completion, the the circle closes. From overstimulation to understanding to restoration to balance. The journey of dopamine restoration. The return to neural silence. The recovery of your biological birthright. Sleep well. Heal deeply. Wake restored.